This video is about the causes of Fragile X syndrome. Fragile X syndrome, named due to a gap in the X chromosome, is a leading cause of inherited intellectual disability. FMR1 is a gene on the X chromosome. Too many CGG trinucleotide repeats on the FMR1 gene silences the gene and causes Fragile X syndrome. FMR1 encodes FMRP, which is an RNA binding protein. Phosphorylation of this protein causes repressed translation of mRNA. This repression is a good thing, preventing CGG repeats, which causes Fragile X. This is a normal FMR1 gene. The gene has 7 to 52 CGG repeats in the 3' untranslated region. These repeats allow for the transcription and ultimate translation of FMRP protein. FMRP regulates transcription by binding RNA, which is needed for healthy development. This is a Fragile X gene. It has a longer 3' untranslated region. This is because there are more than 200 CGG repeats caused by a mutation. The CGG repeats cause hypermethylation of the gene, which silence FMRP. This means that transcription is not regulated and therefore resulting in Fragile X. The experiment performed by Lee et al. Sought to determine whether FMRP inhibits translation via interactions with the mRNA. The study examined the effect of recombinant FMRP on translation in rabbit reticulocyte lysate. So, FMRP was added to the in vitro translation system with some exogenous mRNAs. The researchers were hoping to determine what effect FMRP would have on the mRNA translation. This experiment tested 3' and 5' RNA strands on translation templates and purified recombinant FMRP. Northern hybridization was used to detect the expression of the RNA. By using immunoprecipitation and immunoblot analysis, the researchers were able to isolate the FMRP protein and evaluate the effects on its affinity to RNA. With increasing doses of FMRP, an in vitro translation process was used in order to show the amount of translation repressed. Lastly, before analysis, a RNA binding assay determined the interaction between the RNA and FMRP. In this portion of the experiment, researchers found that FMRP represses translation of brain mRNA. The greater FMRP is expressed, the more translation repression, which is a good thing. This experiment was compared to a control protein, BSA, which does not repress translation. To expand on this finding, the researchers sought to determine whether the FMRP binding site was necessary for FMRP to have its repressive effects. Researchers investigated where FMRP binds. They discovered it was the 3' untranslated region, or UTR. Without the 3' UTR binding site, FMRP is unable to bind. Next, researchers took things a step further, just as we saw in the last graph. When the 3' UTR is absent, FMRP cannot bind. When it cannot bind, translation is not repressed. The green bar on the left is what is required for a healthy human. You need the 3' UTR to be expressed, as well as FMRP to be present to repress translation. So the take-home message for this presentation is that FMRP is a regulatory RNA binding protein that represses translation. The FMRP protein and the FMRP binding site are necessary for translational repression. Silencing of the FMR1 gene causes a lack of FMRP protein, which decreases repression, which is bad. Without translational repression, certain harmful mRNAs are translated in high amounts, leading to Fragile X syndrome. Thanks, you guys, and good luck on the exam!